Okay, so let's start this last session of the first edition of TraCyTalks about beads. And today we will talk about mainly about small particles, mainly small, so extracellular vesicles. And today, first of all, let me again tell you already heard this from the last sessions, but so it's I'm organizing this webinar, but we always have Alexandre Salvador with us. That always helps me with this. Thank you, Alexandre. And for all talks, if you have any doubt, any questions, any comments, you can put it in the chat. And me or Alexandre, we will take that questions and then we'll try to address in the end of the two talks. So today, to start, we have André Mozes. André is a flow cytometry specialist already for more than 10 years. He started here in Portugal. Actually, he did his bachelor in Porto, I believe, right?
Yeah.
Thank you. And now she is in Lausanne, Switzerland, in the EPFL. So there is the facility there, and he's been involved in projects about extracellular vesicles, and he accepted to be here today. So I thank him for that, and he will talk a little bit about this particular subject in this topic of extracellular vesicles. Thank you so much, André, and I will pass for you the word.
Okay. Hello. Good morning. Thanks for the nice introduction. Of course, I'll start by thank you for the invitation. So Catherine and also Alexandre, of course. Trying just to start sharing my screen.
I think you can share. Let me put here.
Yeah, you are still sharing it. Okay. Let's see if it's the right screen. Is it okay? Are you seeing the next ones?
We are seeing the next ones.
It's the other one.
Like that?
Yeah.
It's okay?
Yeah.
Okay, perfect. Okay, let's start then. Okay, so as Katarina said, nowadays I'm working in Lausanne, and basically when she and Alexandre came to me to give this small talk about EVs, the idea is not really to go into detail about what they are, their biogenesis, and so on. The idea behind this presentation is more of if you are starting a project in extracellular vesicles, especially if you want to do it using flow cytometry, what are the aspects that you should think about? Okay, so basically, what do you have to keep in mind before starting your project on EVs using flow cytometry? Okay, so I'm going to do kind of an overview about some of the points that you should keep an eye on. Okay, so to start with, basically, why are we here today? It's crazy the amount of papers that are being published these last couple of years regarding the extracellular vesicles. Okay, so we can see this exponential growth in the last years. So these are data from last week, quite fresh, and you can see that especially in the last 4 or 5 years, the number of papers is more than 4,000 per year, which means that it's a field, it's really a hot topic at the moment, and a lot of things are being published nowadays regarding extracellular vesicles. Then, of course, the question is, okay, so are they really new? Are they newly discovered? I would say not so much, because the first time that today it would be described as an extracellular vesicle, it was in 1946 by this guy here called Erwin Chargaff. So he was working with Randolph West, and basically they published this first paper where they described them as plate-derived particles. Okay, so nowadays they would be classified as EVs. And well, over the years, some more papers have been published in this. Important papers have been published in this field. So we have some from Harding and Pan, also in '83. Also then in '96, Graça Raposo, working in Paris, was probably the first one that defined exosomes as signal entities, okay? And probably the most important one, or the one that caused all this boom, was in 2006, 2007. So two papers, one from Ratazak and the second one from Valadi, that basically stated that these EVs can contain RNA. And so this information could be transferred between cells in a functional way. So meaning that until now, let's say, people also or always thought that this signaling between cells were only when cells were in contact with each other. And now they describe the way that cells can communicate even if they are not located in the same region. Okay, so the idea behind more or less like this message in a bottle. Okay, so cells can send information from one another even if they are not in touch. So of course that after this publication, a lot more came out. And as you can see, so if you regard the human body, let's say in all kinds of fluidics, these EVs have been described, okay? Also all kinds of systems, even between plants and even lower organisms, okay? So people have been describing EVs all over, so they have been identified in all kinds of species and fluidics that you might imagine, okay? In terms of research applications, also diagnostics, of course, they are being quite used looking for biomarkers, vaccines also, and being used as a drug delivery vehicle. Okay, so these are the main applications that nowadays people are really looking, why people are really looking into EVs nowadays, okay? So in terms of their classification, just to let you know how it started more or less. So people started by describing them or classifying them only based on their size. Okay, so I just need the pointer. Okay, so basically everything that was below 100 nanometers was considered to be an exosome. From 100 to 1 micron was the microvesicles, and then above the bodies, and then you have the cell here to compare sizes. But then some people said, okay, but if they are 98, 95, are they for sure exosomes, or they can be small microvesicles? Okay, so then people came also with the biogenesis. Okay, so then we can combine the size with the biogenesis. So if they are formed internally in these multivesicular bodies, they are considered to be exosomes. If they are formed on the budding of the cell surface, then they would be microvesicles. Okay. And as you can see, after some time, even exosomes, so the size was not even more a way to classify them, because we can have exosomes, let's say from 50 to 150. So those 100 there were out. Also microvesicles, so the size, they can have same size being one exosome and another microvesicle. Then another problem in this field was that there were different names to classify the same type of vesicle. Okay, so even up today, there's not really a nice consensus about what do you need to have to describe it as an exosome or as a microvesicle. Okay, so normally I prefer to call it extracellular vesicles and describe it. Okay, I have an extracellular vesicles with this size and go from there, and not really say, okay, I am able to select or sort exosomes when I'm not sure that the vesicle that I'm working with are real exosomes. Okay, especially because there's no consensus on this classification. You can see that even later on, they started to classify exosomes, for example, a bit like the clothes. So like your t-shirt is a S-size exosome or an L-size exosome. Okay, so it's really hard to give a classification that everyone agrees with. And also what was published last year. So if the size is a bit controversial, the biogenesis was always quite accepted. Okay, as I said before, but last year, so this paper said that we might have some exosomes that are not formed into these multivesicular bodies, but they are released by the budding of the membrane. Okay, something that was only said to be done by the microvesicles. Okay, so you can see that it's still probably a bit too early to really classify or to have a good criteria to say that we are working with exosomes or microvesicles. Okay, and when you see a lot of data that has been published, it contains a lot of mistakes because they don't take in account this kind of lack of consensus. Okay, so of course I know it's much sexier to say that you are working with exosomes, but if it's not true, I would avoid to say such a statement. Okay, but that was it in terms of classification. So, okay, now we know what EVs are, but why should we use flow cytometry to describe them? Okay, there are many techniques that you can use to identify them or select them. So why flow cytometry? More or less the same reasons why we use flow cytometry to analyze cells. Okay, so it's a multi-parametric tool. You can get multiple parameters from the same cell or the same EV in this case. It's a high throughput technique. Okay, so especially if you have some samples to acquire, of course, it will be much easier to get a lot of data using flow cytometry than some of the other techniques. It's also worldwide available. Okay, it's also important. So you'll find a flow cytometer next to you, probably doesn't matter exactly where you are in the world. And if you need any other reason, we can say that just because we love it. Okay, so there's no need to another reason to use flow cytometry. Of course, now a bit more serious. If you compare the most used techniques in this field, and depending how you want to characterize or which is the measurement that you are going to use to classify your EVs, you can see that flow cytometry allows you to have most of the options that you might need. Okay, so of course you can love it, but it's also a more realistic idea behind it. Okay, so then now we know that we are going to use flow cytometry to do our study. And so what comes to my mind when I think about flow cytometry and EVs at the same time? So it's an old picture there. Of course, in any kind of analysis, sensitivity and resolution should be on your mind. So do I have the right equipment to do that? In terms of side scatter, again, they are small particles. So as a friend of mine would say, small particle, a big problem. So probably for scatter, side scatter might be a problem to detect these kind of particles. Also, due to their size, if I want to stain them, they will have a low epitope number. So I have to keep that in mind. Refractive index, we'll see it later. Also, can I distinguish them from my background noise? Let's see. So threshold also, they are quite small, so probably I'll lose them below my threshold, if I still using the forward scatter as the default one, swarm, coincidence events, and then as in any kind of analysis, controls and sample preparation is always the key point in any kind of analysis. So basically, I divided a bit these options or all these problems or things that you need to keep in mind. And so I started with the ones that are instrument related. Okay, so do my instruments have enough sensibility to be able to detect my EVs? Do they have enough resolution? And all the others that we'll see a bit more in detail. Okay, so the good news here is that these are quite easy to solve. Okay, there are new equipments in the market that are made to analyze EVs. Okay, so nanoflow, as they call it nowadays. So it's the easier solution. You just buy one of these machines and you don't have these problems anymore. Okay, this is easy, but if you are in the same case as myself, it's not always accepted. Okay, so if someone like my boss said, okay, no, we are not going to buy one of these equipments, and you have to work with what you have in-house, then you have to make sure that you don't have any problem, or that the machine that you are going to use to do this analysis is in the best shape possible. Okay, so of course here I just put this here because in the first session we talked about these QC beats. So of course all the QC should pass, CVs as low as possible, of course. In terms of scatter, what should I think? Do I have the best filter in my equipment to detect it, to detect low scatter particles? Is it possible to get the side scatter from another laser, like the violet laser, rather than from the blue one? And if it's possible, is it better in my equipment? Or even if it's possible, I still have better results with the blue side scatter? And of course, if I have a lot of problems, or if my equipment does not have that sensitivity, probably why should I use beads? Okay, there are tons of beads. Next presentation will be about beads and extracellular vesicles. So they are a great way to overcome these scatter problems. Okay, of course, if you use some beads to attach to your particles, then you will increase the overall size and you will avoid most of these problems. Okay, so if beads is possible to match with your downstream application, so why not? And then regarding your instrument noise, so here I stated it as cleanliness. For me, is basically the key points when you try to analyze EVs. Okay, and what do I mean by that? Basically, everything should be as clean as possible. Okay, in terms of buffers, sample buffers, all the reagent that you use, if you stain your samples with a fluorochrome or something like that, everything should be filtered. Okay, otherwise it will contribute to increase the background. Also the instrument, what type of sheaths does the instrument uses, PBS, what kind of PBS, even sometimes some PBS commercial ones gives a bit more background than others. Okay, so test a couple of them, or water, depending on the machine that you use. Also should be filtered at, so we use 0.04, okay, to filter all the buffer. If you use, or if you have a spare fluidic tank, for example, we have that in our equipment. So we have one fluidic tank just for EVs, okay, or small particles if you want. Okay, so that's also a good option, depending if you have it or not. And then flow cell, of course, as clean as possible, and also all the sample lines. Okay, so just to give you an idea, I said here the strategy or the cleaning protocol that Joanne Lannigan uses. So it's one of the people that works a lot with EVs. So just for you to see that she does at least 3 long cleans plus the startup before running any tube. Okay, so for those people that say, okay, the equipment is free for half an hour, I'm just going there to acquire a couple of tubes, that doesn't work. Okay, that half an hour you will need just to clean the machine to get good results from the tubes that we'll acquire later on. Okay, so I would say at least this half an hour cleaning is mandatory if you really want to lower the background on your analysis. Okay, because if you go there and the machine is not extremely clean, it doesn't work for your EVs. Okay, so if you have to book the equipment, I would book probably 1 hour just to clean the machine, or at least half an hour, and then the rest to acquire the tubes. Okay, so there's no way you can do an acquisition in half an hour. Okay, cleaning is really important in this case. Then other features or that are related more to the acquisition. So in terms of refractive index, just to let you know, so most of you already know it for sure, that if you say, or if you compare 100 nanometer beads, for example, with the same size particle. So if you say that, for example, my beads that are 100 nanometers, they give this amount of side scatter, for example. If my particle has the same level of scatter, it has the same size. Okay, so that is not correct because they don't have the same refractive index. Okay, so they will be seen much lower than those kinds of beads. Doesn't matter if it's a polystyrene bead or silica beads. The scatter from your EVs are going always to be smaller in terms of scatters. Okay, that's also something that you have to keep in mind. In terms of swarm detection or coincidence events, so the same as you try to avoid this regarding your cell analysis, with EVs, it's much more important, okay, because the EVs are much smaller than your laser beam. Okay, so what you want to avoid is having more than one EVs being analyzed at the same time. Okay, so how can you do that? So here you can check, let's say, the relation between the count and the concentration. So if you have a nonlinear relationship, means that you are having some swarming effects. Also, of course, through serial dilutions, you can do something like this evaluation. Okay, so the fluorescence is kind of stable, while of course the counts are being decreased when you dilute your sample. Okay, so also really important, try to avoid these coincident events. Okay, and then when you have to set the threshold, of course, if your particles are fluorescent, that is much easier because you can set the threshold on the fluorescent marker. Okay, if they are not, and if you have to set it on forward scatter or side scatter, make sure that with the value that you set, probably the default one is always too high. Okay, make sure that you see the bits or the particles of interest. Okay, so just as a rule of thumb to check if your equipment is more or less clean, if you set it on the forward scatter, side scatter, you should have below, it should be below 200 events per second, just acquiring buffer. And if you have a threshold on the fluorescence, normally it's about something below 50 events per second is considered to be clean. Okay, of course, lower the value, better it is for you, but it's just for you to have a reference in these kind of values. And then again, if you want to stain them, okay, don't forget that they are much smaller than your regular cell. So for sure, choosing a really bright antibody would help you then detecting them. Okay. And then, of course, something that is always present in your flow cytometry experiments are the controls. Okay. And to show you that they are really important, I know everyone says that, but so basically this was a survey that was sent by this group, the MyFlow Site EV. So they are a group of guys that work with EVs, And you can see that most of the people that answered are reviewers from the main journals that links flow cytometry and EVs. Okay, and you can see that basically this blue bar here, as big as it is, so it makes more consensus for these reviewers to describe that whenever you submit a paper about EVs. Okay, and you can see that the controls are basically important, or they are considered to be important by many of these reviewers. Probably the one that is not that important, also not in my opinion, I would say, it's this isotype control. Okay. Apart from that, all the others are basically mandatory if you want to publish something in this about EVs in one of these papers, in one of these journals. Okay, so then these are basically at least the minimum controls I would say that you need to have. So buffer only and buffer with reagents, it will give you the background level. Okay, so if you don't know your background, you have to have it, otherwise you don't know where to place your gates, for example. Okay, then the unstained control. Okay, some EVs have autofluorescence and even different autofluorescences, so it's always good to have an unstained control. Okay, to especially set your gates. Of course, if you use more than one color, you might need to compensate that. So use single stain controls. Also to see background contribution in other channels. Okay, you can also use these single stain controls. And something that is also being asked nowadays are these procedural controls. Okay, so basically the idea here is to show that, for example, if you use another method to isolate your EVs, for example, if you use ultracentrifugation, so to make sure that that protocol does not bring any background to your acquisition. Okay, so your controls, basically these ones before, should follow the same procedure as your samples. Okay, to make sure that is not your the method that you did before that gives more background to your analysis. Also important, I think it's quite easy to understand, but all the controls should be acquired with the same settings as your samples. And normally what people do is to set the stop criteria on time, okay? Instead of setting it by number of events. If you use time, so basically, in that case, you are sure that the background contribution is constant through all, okay? And it's much easier to explain and to make sure that you have the same background contribution. If you want then to report and to compare results with other labs, for example, it was also asked on this survey, and you have to make sure that if you give any concentration values, that your volume is well calibrated. Okay, so here also depends on the equipment that you are using. If you have a syringe pump, make sure that it's measuring what it says it's measuring. Otherwise, if you don't have it in your equipment, you can use that mass discharge method. Let's say basically it's by weighting the sample before and after acquisition, or use this true count bits. Okay, you also get the accurate concentration of your sample. Also, in terms of fluorescence, okay, so basically you need to report the fluorescence level in a quantitative way. Okay, so if you check the value that you have on your dot plot, that one is an arbitrary unit. Okay, so you have to convert it into a quantitative one so that you can report the fluorescence, and that can be basically compared to other labs. Okay, instead of having an arbitrary unit, then you have a quantitative one. Okay, for that, as we are talking about beads, you can use quanti bright beads, or this quantum from Banks lab also works quite well. And then something that you also have to calibrate is the scatter. Okay, especially if you are trying to say the exact size of your vesicles. Again, like the fluorescence is arbitrary. So using this me theory, for example, if you have some particles with known refractive index and also known diameter, you can calculate this calibration factor and then apply it to your EVs. Okay, so if you have then your scatters well calibrated, then you can report the exact, or at least the most correct size as possible. Okay. So I also set some literature here. So I think these are at least important if you are starting your project. Okay. So many of the these issues that I talked about are described in these papers. Okay. And I think it's also a good idea to read them before really starting planning your experiment. It will save you a lot of time and a lot of problems. And my time is almost finishing. So as a conclusion, and to answer my title, I would say that yes, so we can analyze submicron particles or EVs by flow cytometry, but for sure it's challenging, okay? And you need to control every step, okay? So again, you have people working in your core facility. So basically go discuss with them, ask them which is the best equipment, what can we do to improve results, because you might know the biological part of it, but they know the technical aspects, okay? And we have seen it in our facility, some groups that tried by themselves and some that started already worked with us. And we can see that the ones that worked with us, they go much faster than the other ones. So the other groups, normally they try it, and then they try to publish and is not accepted, and then they come to us and ask us why. Okay, so then they have to go back and start it over. Okay, so I think it's a nice message to contact your platform first and then discuss with them what you want to do, and it will be much easier and much quicker. Of course, a couple of acknowledgements. So from the people that are working with me in the facility to get me some spare time to work with these EVs. People from different groups that have worked with me and with the facility on this subject. And then also Joanne Lanningen, that was in the University of Virginia. Now it's working for SciTech. But yeah, she's also a good person to ask if you have some questions in this field, and I'm sure she will be happy to answer you. And of course, thank you very much. And I'll leave my email in case you have some questions. Thank you.
Thank you, André. Thank you for your presentation and these tips and tricks to analyze IVs. I think it was really important. I still don't have any questions or comments in the chat. I'm not seeing nothing. I don't know if you kept something from there, Alexandre. No microphone, Alexandre. You don't have microphone.
No, I don't. But I have some questions too.
Okay. I was saying that maybe if for the participants, for everybody, if you in the end want to, you have questions or comments, even for André, I think André won't mind that if some questions are addressed to you in the end of it's okay.
Sure. Yeah.
But if you want to do some comment like Alexandre now, feel free as well. Or
no,
use the microphone.
No, it's just some small questions, because I was looking at some of the work that André has been doing in this field, and I wanted to ask him if he wants to share with us some of the first, for example, experiences that he had. For example, I saw that there are some comments in the web saying that, for example, only with the violet side scatter you can do EVs. Also that you really need a dedicated instrument for the analysis of the EVs. And I know that André has been doing quite a work doing a different instruments, even including cell sorting. So can you just comment on that, saying, is it possible or not? What are the most difficult challenges and how did you overcome them?
Yeah, sure. Thanks for the easy question. No, I'm kidding. No, it's true. So basically, of course, people state I also started using the side scatter from the violet laser because it's basically what you see published everywhere. So the side scatter from the violet gives you always better results than from the blue. And in my opinion, it's not true. So actually the equipment that we use the most with EVs, we use the side scatter from the blue laser, okay? Of course, people have to keep in mind that when they say that the side scatter from the violet is better, it also depends on the laser power that you have on that laser. So if your violet laser is much weaker, for example, than your blue laser, most probably you get a better discrimination or sensitivity, if you want to call it like that, from the blue laser rather than from the violet one. So I think those kinds of dogmas are hardly ever correct. So it really depends on the equipment that you are using. And yeah, I can assure you that we can use the blue laser to get or to separate and identify your extracellular vesicles. Okay, it's not mandatory to do it from the violet one. What I normally say is that if you have the opportunity, just test them. Okay, you can test them in parallel with the same sample and check from which laser do you get the best side scatter. It's as easy as that. Of course, if you have different equipments, then you can do that comparison into or using different equipments. Then if you need to have a dedicated machine, of course, that would be the ideal. Okay, I think it's more or less in the same level as buying an equipment just to do extracellular vesicles, those kinds of equipments that I showed before. Of course, if you have the money, if it's really a huge subject in your lab, why not buying one that is dedicated to that? Of course, you cannot run cells on those equipments, but if you have enough work with EVs, why not? But we know that realistic, hardly anyone can do it. So yeah, you can use any kind of flow cytometer. Then the idea for me is it really needs to be well cleaned and well aligned and so on. Okay, so in your case, like in ours, as we have staff in our facility, it's much easier to make sure that the equipment is well aligned and so on, and even cleaned. That some labs, for example, that have their own equipment and they don't do quality control of their equipment, for example. Everyone knows cases like that. So it's much easier when your equipment is controlled daily, and then it's up to you to make sure that it's clean enough, because you never know what the person before you did, what kind of analysis the last user did. So for me, yeah, I don't run any tube without cleaning it for half an hour. So it doesn't matter which equipment you use. If it's not cleaned, you will not see your vesicles. The background will be too high. Then regarding the equipment, yeah, there are many equipments where you can do that. If you follow these rules, so here the idea was not really going into detail in all of these things or subjects that you need to keep in mind. It was basically just to talk about them. And then if you are going to work in this field, you can review the presentation and see, okay, this guy talked about, I don't know, threshold, for example. So which one is the best for me and so on. And yeah, I think it's possible to use different equipments, even cell sorters, as you were mentioning. It's really hard. It's really a long sort. It takes a lot of time. Also to avoid the swarming effects, okay, it's really important to acquire your samples as low as possible, as I would say, as diluted as possible, if it's something that we can say like that. And of course, if you need to sort a huge amount of particles, you need to sort for a long time. So long days in the lab, yeah, for sure.
And just one thing, and the cleaning for the sorting is the same time? You use the same time cleaning? So only the half an hour, 1 hour, or it's even more time when you go for sorting.
No, I did the same because especially at that moment that I was more interested in sorting them. I had the equipment just for EVs. So it was always more cleaned than the normal sorter. But yeah, at least half an hour I would do it
please.
anyway, yeah. Okay,
thank you so much, André. I don't know if we will have more questions. Even me, I'm curious about other topics, but I think we should proceed.
Sure.
And thank you. So the next speaker is Ricardo Jara. I think he's somewhere. I think he's here. I'm sorry. So it's Ricardo. Ricardo is one of the associates of Immunostep, and is not only one of the associates of Immunostep, is also been involved in a lot of techniques, so in developing new techniques and reagents, then to be applied in research, and of course in another research and sorry, I don't know, too many tabs. Good. Okay. So he's one of the associates of Immunostep, and he's been involved in developing reagents and techniques, then to be applied in the research area, and he's been collaborating as well with directly with groups in the researching. So I need to thank him so much for accepting to be here today. He will talk a little bit so how to use these beads to capture beads, then to analyze extracellular vesicles, and then to analyze them by flow cytometry, as you can imagine. So thank you so much, Ricardo, and I will. And now I think you can share your screen. No. Okay. Okay, now we can see. Thank you so much, Ricardo. No. Yes. We are not hearing you. You need to turn on the microphone, please.
Now?
Yes. Now,
okay.
Yes.
You can see the presentation?
Yes. Yes.
Yeah. Okay, perfect. So, first of all, good morning, and thank you to you, to Katerina, and to Alex, and to the organization for the invitation to present this work, Immunocapture-Based assay for detecting extracellular vesicles by conventional flow cytometry. Oh. I have a problem.
Again, it's the slide that is not moving, right?
Yeah, it's not moving. I don't know what's the reason.
Here.
Let me see.
It should be flowing, not static.
Okay. Maybe I can ask Danielle. I don't know if Danielle is here.
Yes, hi.
Yes.
I would love to help.
Let me see. I'm going to let me see.
It seems to be a problem within your own software.
I don't know. Let me see.
Meanwhile, just to make a comment, Ricardo is doing some of this work using a similar technique that we have described last week, okay, with some captured beats. So they developed some of this work in collaboration with some universities in Spain.
Yeah. You can see now the presentation.
Yes.
Yes.
Yeah.
Yeah.
And it's moving, no?
Yeah.
Yes.
Okay, perfect. Sorry, I don't know what's the problem, but you know. So now, just to say thank you to organizer to let us to present this work, titled "An Immunocapture-Based Assay for Detecting Extracellular Vesicles by Conventional Flow Cytometry." As you know, most cell types release extracellular vesicles during physiological process, and the term incorporates microvesicles and apoptotic bodies and microvesicles, and the term which differ in their site and origin, and the term of exosomes include nanovesicles with a diameter between 30 and 150 nanometers, and you can also find them named such as small extracellular vesicles. In this presentation, when we are talking about extracellular vesicles or EVs, we were talking about small EVs or exosomes. As you know, also EVs are released by most of mammalian cells, reflect the state of the precursor cell at the time exosome generation. This is quite important also, and are easily available in nearly all body fluids and have an endotheltic origin. But which means that they have an endotheltic origin. Exosomes are generated by the fusion of the specialized endosome, the multivesicular body, with the plasma membrane. Specifically, the generation of exosome occurs through the invagination of the plasma membrane to form the early endosome, and subsequently the inward budding of the endosomal membrane generate a multivesicular body that can follow two different routes. Go to the lysosome for the content degradation, or fusion with the plasma membrane to release the exosome. This process, named exosomal biogenesis, distinguishes exosome from the rest of extracellular vesicles, since the largest microvesicles, for example, are produced by budding and fission of the plasma membrane, while the apoptotic bodies are released as blisters when the cells suffer apoptosis. In addition to the particular biogenesis, exosomes are also unique in their molecular composition at the level of proteins, lipids, and nucleic acids. And as well, as we mentioned also before, this molecular content is a reflection of the state of the precursor cell at the time of exosome is generated. For example, highly abundant cytosolic proteins, including metabolic enzymes or cytoskeletal proteins, are missing from exosomes, which means that the uptake of the components during exosome biogenesis is not random. It's a highly regulated process. In this sense, all the exosomes have proteins involved in the exosome biogenesis, also proteins associated with endosomes, integrins, and of course the traspanin, CD63, CD9, CD81. On the other hand, several classical biomarkers for exosome, like major histocompatibility complex, has or even shock proteins have more or less the same present in all EVs. And regarding the nucleic acid, it's not possible to find, but it's not very clear, but I think that it's not possible to find doublet strained DNA. However, we can find extracellular RNA, such as microRNA, although exosomes don't carry the necessary molecular machinery to carry out the cell-independent microRNA biogenesis, like, for example, Argonaut proteins, for example. And from the lipid point of view, probably phosphatidylserine is not present in the outer leaflet of the exosome membrane, for example. And finally, I think that it's interesting to come back to the tetracycline, because these proteins are considered a pan-EV marker, so it's common for most of EVs, and are used frequently as markers for exosomes, for exosome identification, for exosome-specific identification. So worth highlight that the importance to carry out the combined analysis, because both CD63 and CD9 can be present alone in other type of EVs different from exosomes. As you probably know, in the recent years, exosomes have generated a great interest. And why is this explosion of interest? I say that it leads for three reasons. The first, EVs, or extracellular vesicles, provides a new means of intracellular communication and transmission of macromolecules between cells. For instance, exosomes can mediate and regulate the immune response dependent on major histocompatibility complex, specifically exosome derived from B cells and dendritic cells. All exosomes spread bioactive molecules as contributing factors in development of several diseases. Thus, it's well known the role of microRNA exosomes content in regulating metastasis, angiogenesis, or chemotherapy resistance, or the significant role also that have in the transmission and propagation of peptides or protein aggregates involved in the neurodegenerative disease. The third reason for this explosion of interest is that exosomes are useful tools as vectors for drugs, as active substance for cell therapy. The exosomes are composed of cell membrane rather than a synthetic polymer, for example, and therefore better tolerated by the host, by the human body, for example. At the same time, that reduces the exposure of the content to the host, to the proteases of the host, and to the antibodies of the host. And additionally, has the ability to cross also the blood-brain barrier, allowing the cargo delivery in the nervous central system. Finally, mesenchymal stem cells, derived exosomes, has a great potential for regenerative and cell therapy. Now, let's take a look to the current practice for isolation and characterization of this kind of EVs. With respect with the starting material for EVs isolation, the most common body fluids employed are plasma, serum, urine, and probably milk. Regarding the starting volume by sample type, the majority of the researchers that report a starting volume higher than 100 milliliters used cell culture media, whilst those researchers that report a starting volume lower than 1 milliliter studied mainly biological fluids. About isolation methods, ultracentrifugation remains by far like the most widely used primary method, followed by a combination of isolation techniques. However, when the starting volume is small or a sample corresponds to a body fluid, researchers employ more frequently methods like precipitation or magnetic beads or side exclusion chromatography. Concerning the characterization methods, the three most widely used are Western blotting, single particle analysis, and, for example, electron microscopy. And in most cases, isolation and characterization of EVs has a subsequent downstream application, where the most common are in vitro functional assays and RNA analysis or also proteomics, maybe, among others. However, the diagnostic use of EVs derived from pathological cells has been technically limited by the lack of reproducible and highly sensitive methods. And for that, we think that bit-based cytometry could play an important role in this area. To establish the conditions for a high method, a method of EV immunocapture, we first considered which parameters are critical to get the best signal-to-noise ratio in a flow cytometer. It's known that there is a correlation between the fluorescent intensity and the amount of marker per cell, of course. And so for optimal signal detection, at least two parameters need to be carefully established. The first one is the number of exosomes that contain the marker used for the capture. And the second one is the total number of bits per test. Keeping in mind that relevant exosomes can be in limiting amounts in biological fluids. For a better understanding of the system, we estimate the maximum binding capacity per bit. This was calculated assuming an exosome average diameter of 150 nanometers. And according to these calculations, as 6 microns bits would be captured a maximum of 6,420 exosomes. And if we employ 3,000 bits per test, we can capture up to 19 millions of EVs per assay. This is more or less our estimations. In this other side of the slide, here we can see a schematic representation of the bit binding cell, in which exosomes are captured by the magnetic bit, magnetic and fluorescent bit that is conjugate or coated with the one antibody. In this case, is CD63. And you can use different antibodies for the detection. In this case, for example, CD9 and CD81. This is more or less the idea of the system. So, to confirm the specificity of the recognition the specificity of the recognition, we carry out the signal comparison between microbits conjugate with IgG1 isotype control and CD63 using EVs derived from human prostate cancer cell, named PC3. And for the capture, both types of bits were incubated separately, and the detection was carried out with a biotin-related antibody, in this case CD9. Getting a stronger signal for CD63 and no signal for the IgG1 bits. Also, antibody blocking experiments were also performed, including a sample pre-incubation step with the same purified antibody used for the immunocapture, and with 50 nanograms of these antibodies were sufficient to completely block the binding of EVs to the both types of microsphere. In this case, microsphere bits conjugate with CD9 or with CD63. Additionally to the to this specific comproval or testing, we optimize the condition for incubation with different volumes, temperatures, and times. And the result was that the better condition was incubation without shaking, room temperature, and overnight incubation. So in this way, the protocol will be as follows. The magnetic beads are incubated with a sample at a room temperature without shaking and an overnight. After incubation, the beads are washed on a magnet, because if you remember, the beads are magnetic. And then after the wash step, you can proceed with the incubation with the fluorescently labeled detection antibody. When you're using biotin-related antibody, an extra incubation step is necessary with, for example, fluorescent streptavidin or other secondary reagent. And following the last washing step, you need to resuspend the beads in 300 or 350 microliters for the flow cytometry analysis. For the acquisition, we follow the next simple strategy, a first gate on forward side scatter dot plot. And this region corresponds to the 6 microns beads and help to remove debris and aggregates. And a second gate in, for example, APC channel to confirm the presence of the beads. As we mentioned before, CD63, CD9, and CD81 are the general markers for EVs. However, these tetraspanins can be present in different amounts on EVs subpopulations. For these reasons, the first time you work with a specific EV or from a particular cell line, it's recommended to characterize EVs using standard methods, following the recommendation from the, for example, International Society of Extracellular Vesicles. And this will allow you an estimation of the number of EVs released, in this case from the PC3 cell line, the prostate cancer cell line, by nanoparticle tracking analysis, and as well as identification of the protein that are relatively more or less abundant in these EVs by Western blot. In this assay, for example, we employ non-reduction condition for the tetraspanin, and the exposure time were 1 hour for CD63 and 5 minutes for CD81 and CD9, which give us an idea of the marker expression. In this way, we determine the optimal capture detection tetraspanin antibody combination as follows. The CD9 and CD63 conjugated magnetic beads were incubated with the PC3 extracellular vesicles or without vesicles for the negative control. And after incubation, the sample preparation were stained with either CD63, CD81, and CD9, fluorescent antibodies, of course. And also we keep constant the number of EVs in all these assays. And the results show that although CD63 was in general a less abundant protein than CD9 or CD81 in these vesicles, according to the higher time of exposure required for the Western blot experiments, the CD63 antibody was suitable for the capture when either CD9 or CD81 were used for the detection. In contrast, this is these two histograms. In contrast, CD63 was not as efficient when used as detection antibody. Finally, to confirm that ABs capture on magnetic, or to confirm the EV's capture on CD63 magnetic beads, the sample preparation was analyzed for transmission electron microscopy. This is the surface of the beads, and these gray dots are the exosome, and these black dots correspond to the salt, to the iron salt, because the bead is magnetic. Next, to determine the dynamic range and the limit of detection of the Assay, the EV titration experiment were performed using CD63 and for capture and CD9 for the detection. The sensitivity of the Assay was very high, with a positive signal detected as little as 30 nanograms of exosome, while 2 micrograms were required for the Western blot experiments, for the detection in Western blot experiments. Besides, cytometry data follows a linear distribution between 30 nanograms and 8 micrograms of exosomes, allowing interpolating fluorescence intensity values to estimate, to have a semi-quantitative analysis and to estimate the amount of exosome in the sample. And in this sense, the minimum amount of exosome necessary for a reliable quantification is 250 nanograms. That comes defined by a Stein index higher than 1. And also, finally, different amount of exosome were tested on GG1 beads with minimal increase of fluorescence. To confirm that the EVs from different sources could be detected using this method, we employ EVs from different cell lines. Previously, from different cell lines, but the exosome were previously enriched by sequential centrifugation from the cell culture supernatant. The different cell lines, despite different amounts of CD63 and CD9 as detected by Western blot. However, the detection by flow cytometry with the immunocapture method was possible for LOB10 and with higher sensitivity than Western blot. In this study, for example, EV's detection from several cell lines such as NCF7 or RT112 or SCAML 147, MAMA-MEL 55 or SCAML 28 cell lines could be possible, allowing to correlate the Stein index value with the band intensity detected by the Western blot, also in the same sample. In this case, the band of the CD63 and CD9 was lower in SCAML 147, but despite these finded bands, it was possible the detection of the exosome by flow cytometry. Well, so the experiments above show us a very high sensitivity for the detection of EVs after pre-enrichment by ultracentrifugation. And we hypothesized that it would be possible to implement the assay in body fluids directly with previous enrichment or isolation of EVs. In this case, the CD63 capture beads with the CD9 antibody for the detection, the combination of CD63 capture beads and CD9 antibody for the detection was used first with purified exosomes by ultracentrifugation. In this case, from 30 milliliters of urine. I think that was 30 milliliters. And after, for a direct detection of exosome on just 50 microliters of urine. And in both cases, we use IgG1 beads such as a negative control. And the result showed that in both cases, we have the ability to detect exosome in a small amounts of sample proficient, in this case, urine. On the other hand, when you work with urine, most experiments was pretreated with meal reduction to eliminate the Thom-Horsfall protein aggregates, but we could detect EVs in urine samples in which only the cell debris was eliminated by a simple centrifugation. All these results show that the capture antibody does not need to recognize an abundant protein for efficient capture of EVs, which imply that all kinds of different markers could be studied using this methodology. On the one of the most promising areas in the field of EVs consists in their use as tumor biomarkers. And to prove this hypothesis or this potential use, we decide to use EPCAN, a common marker found in epithelial carcinomas, in combination with the transparins. Firstly, the PC3 derived exosomes captured with CD63 were stained with EPCAN detection antibody conjugate in 5-quercetin, if I don't remember bad. And at the same time, we use captured bits conjugate with EPCAN and detect the exosome with the CD9 conjugate in 5-quercetin. Both capture detection combination allow the detection of EPCAN positive exosome with a very good sign. The specificity of the EPCAN-coated bits was tested in blocking experiments using the PC3 exosome derivative, and by comparing the signal also obtaining with EVs from a EPCAN negative cell lines such as SCAML28. Finally, the expression of EPCAN was analyzed directly in urinary exosomes from 9 donors with prostate swelling or suspect cancer. Exosome isolated by ultracentrifugation from 8 milliliters of urine and analyzed by Western blot and by Flow Cytometry. In this case, by Flow Cytometry, we employ 500 microliters of sample. And EPCAN bits for the capture of the exosome and CD9 for the detection. And the expression of EPCAN was detected in 7 of 9 samples by cytometry, whilst only in 6 samples these markers was detected by Western blot, confirming the higher sensitivity of this method. To finish, in another different experiment, metastatic melanoma derived EV were enriched from mammal 86C cells and analyzed by Western blot to confirm the presence of the EV markers like CD9, CD63, CD81, and other specific markers such as MICA, present in tumor cells, or the melanoma antigens tyrosinase or melanin. The protocol for immunocapture and staining was the same that we have seen before. EVs can be captured in parallel on different types of bits conjugated to antibodies against the protein of interest, CD63, CD9, MICA, PD-L1. And once captured EVs can be stained with the fluorescent antibody, usually direct against an abundant EV markers such as tetraspanin, in this case is CD81 conjugate with 5-quercetin, for the detection by flow. Sample acquisition can be performed in any conventional flow cytometer. This is important, allowing the same gating strategy previously shown, a first gate in forward side scatter to select the region and to eliminate debris and potential doublets, And after that, the confirmation of the bit in APC channel. Once the bits have been acquired, a simple analysis is possible. Just selecting an histogram for the channel detection, in this case, phycometry, and comparing the different samples. Once again, to confirm that these bits capture specifically the melanoma extracellular vesicles, the sample was processed for transmission electron microscopy. And once again, you can see here the surface of the microbit, and the gray dots are the melanoma EVs. This is more or less the confirmation that the specific recognition and the capture of exosome with this methodology. Just a brief summary of the presentation. We have presented a robust and reproducible method for immunocapture of EVs with a high sensitivity detection by conventional flow cytometry. The method allows EVs immunophenotyping, even in small amounts of biological samples, and even the direct detection of EVs from minimal proficient biological fluids, allowing possible the detection of non-abundant proteins on these EVs with a better limit of detection than Western blot. And the method is easy to implement, allowing a larger screening studies. Just a little piece of publicity. This method is commercially available under the name of Immunostep by Immunostep, and I invite you to visit our website where you can find more information and a wide range of different products for exosome field, which are distributed in Portugal with our partner NC Pharma. And that's all. Thank you for your attention.
Thank you, Ricardo. Thank you so much for your presentation. And in the end, it's publicity, but it's also information, and we need that. I think it's the purpose of this webinar. So thank you so much also for that.
I try to do it.
I'll try to. So I don't know if there is any questions for Ricardo, for André, me, Alexandre, we are all here. You can use the chat, you can turn on your microphone. So we have one here from Joana Rodrigues, do you have any tips in how to choose the right flow cytometry method, direct FCM or bit-based to use in our experiments? Maybe it's for me, but I don't know which experiments are we talking about, Joana. Do you want to turn on your microphone? Maybe it will be easier.
Okay. Hi.
Hello, Joana.
Hi.
Yes. Yes.
Okay. So I was just wondering, because today we saw two methods of trying to analyze EVs, and I was just wondering if one is better than the other, how to choose the one to use in our experiments, and not any experiment in particular. It's just, does any method have an advantage compared with the other?
Yeah, I can comment on that. So yeah, so at the idea is that the method is always the same. Okay, you are always using flow cytometry to analyze or to identify your EVs. Then it's a bit more regarding your project. So I think both of them have advantages and disadvantages. So probably, can you use beads, or do you need beads to detect the EVs using the equipment that you have? If yes, why not? Do you want bulk isolation of your EVs? So I would use beads, for example, in that case. Or I have such an equipment which it's so specific and I can detect my EVs. I only need to detect them, and I'm able to do that without beads. Probably I would do without. So it really depends on your particle of interest, on the equipment that you have available, on your downstream application. So it's hard to say which one is the best. So both of them can be used in parallel at the end.
Let me make a comment on that also. First, trying to reply directly and not going around with one. If you're trying, for example, has a starting method, okay, to do the identification, and you don't have available, for example, a good electron microscopy or easy way to do Western blot to do all this validation, because it's important. You want to avoid, for example, a lot of steps of ultracentrifugation and so on. It should be important to have a very clear method. And for example, Ricardo has shown us a very clear method to do this. The kit brings everything that you need. Also, one thing that is quite important is the standard. So most of the times you need to have a reliable result. So the standard included in the kit also can give you that. When you are sure in what you are measuring and seeing, then you can, like André was saying, then you can move more confident to a way of detecting directly, probably your exosomes or extracellular vesicles. Most of the times, I think the difficulty is when you have a sample to be sure that what you're seeing is really extracellular vesicles. Most of the time, the reproducibility of the result when you're doing a direct measurement is very difficult because, for example, like André was saying, you don't encounter the instrument in the exact same way each time you're using it. So most of the times you're measuring more debris than extracellular vesicles. With the use of beads, you can get out of that difficult area that André was saying. So in most of the cases, like Ricardo was showing, when you want to go to a biological sample like urine, for example, very easy to obtain in most of the samples and patients, very easy, because I think that there is a lot of sample per day that you can acquire from patients in this way. Probably it can be easy to start in those kinds of samples or in cell culture. Cell culture, one of the difficulties is when you have adherent cells, can be a problem because you use other techniques to do the isolation. Then you don't know if you're talking about, for example, the leftovers that you have there in the supernatant, or if you're looking exactly in what you want to measure, the way to communicate between cells. So I think that to be confident in what you're doing, instead of using, for example, a Western blot that takes a lot of time, doing also this, and I think that Ricardo can talk us a little bit about this, but doing electron microscopy also in extracellular vesicles is not simple. There are a lot of artifacts in all these techniques. And I think that, for example, the bead assay can be a very, very easy way to start once you want to address the extracellular vesicle techniques. If you start by something that you have standards, you have all the solutions, and you have the phenotypic approach and the size approach, I think can be an easy way to start, okay? Because it's not easy, for example, like André was saying, to get people with enough knowledge and experience to help you out once you want to start with the extracellular vesicles. So I don't know if Ricardo has something else to add, but I think that this will resume a little bit.
No, I completely agree with your comments. Of course, when I showed you in the presentation transmission electron microscopy or Western blot, the idea is that probably we can do this kind of comparison for you. So this is if you can implement an easy method in your lab for a big number of samples, looking for example, biomarkers. Probably you need to put the focus in the samples and the data, the analysis, the high throughput analysis. So it's impossible to make a transmission electron microscopy or Western blot for every sample. So the idea is, okay, you have a sample method for a conventional flow cytometry with a not too long protocol. I mentioned that we use a overnight protocol incubation for the sample, but we have results also with 2 hours of incubation with the samples. But the idea is to have a better signal with overnight incubation. But it's possible to reduce the protocol to optimize with the method the sensitivity of the technique for your samples, and to try to take all the complex setup around the samples, around the data, the clinical data, and forget the setup, the method, the transmission electron microscopy, the Western blot. That's true that if you are planning to make some kind of publication, probably at least one time it will be interesting to have another kind of characterization of your samples. With a regular sample, you make the Western blot, the nano tracking particle analysis. I don't know if it's possible electron microscopy, but you can follow the recommendation of the International Society for Extracellular Vesicles, but probably it's enough to make this analysis with just one sample or several samples. After that, you put the protocol. You can run the protocol with a simple analysis by flow cytometry with this method.
Yeah,
so I think that what André was also showing is that, for example, the kits are the kits and have only some antibodies and some approach. Once you want to go through your imagination and start researching other things, then you have the method validated, and then you can move with the flow and do other things, and very interesting things, and test other markers and so on. That's why I think André was saying that in those cases, if you want to move after to all these techniques in flow cytometry, there is a big field in there. There is a lot of approach that you can do also.
Yes, but maybe to start, it is a good idea to start with capture bits if you can.
Just another question. We have no show data about that, but the bit method is compatible with the downstream protocol for microRNA analysis, for example. You have enough sample in order to make the capture, the check or the comproval by flow, and after that to analyze the captured exosomes by molecular biology in order, for example, to analyze microRNA content of these captured vesicles. This is completely compatible. It's not necessary to evaluate or to remove the bits for the downstream protocol for microRNA analysis.
Yeah, thank you. I think that is a really nice information. Joanna, I don't know if they addressed your question.
Yes, it was really helpful. Thank you.
Okay, so anybody has any more questions? You can also, I'm hearing a sound, but I don't know. Okay. Any more questions? You can use the chat, microphone. No, it is clear. You have, Alexandre? You have a comment, question? It was just yours.
Oh, my phone. It was my phone ringing. Sorry.
I get it now.
I was not giving you any music in particular, making it more fun. Sorry. Thank you for that.
So,
sorry, can I just make a question?
Yeah, sure.
Hello.
Yeah.
I'm André, I'm a researcher in here. I was wondering, Ricardo, can you use more complex samples from more complex matrices? Like, for example, you gave the example of urine. Could you use with more complex matrix, or this would give like artifacts of unspecific binding, and then you would lose the capacity to detect your EVs?
Ricardo.
Yeah, sorry. Good question. No, I showed you just that about urine and cell culture and so on. But of course, the method is completely compatible with more complex matrices like plasma, serum, or other thing. In fact, we have several different kits for different samples. We have a specific kit, for example, to work with plasma or with serum. And you can also use the kit for a pre-enrichment sample from plasma, for example, using site exclusion chromatography columns. But you can also to try to put to work with the direct sample in plasma. For example, here in Madrid, we have a couple of clinician groups working in cancer using directly plasma for screening of lung cancer, for example. So that's completely possible. It's not necessary to use just urine or cell culture media. The methodology works properly with plasma, for example.
Because I was just thinking on, for example, when you have a less complex sample, like for example cells, supernatant or whatever, I would think that the exosomes will have, or the extracellular vesicles in this case will be exosomes, would have to have a tendency to be more homogeneous. For example, if you have highly fluctuable expression of your marker of capture, for example, you have cells that express more exosomes that can have reduced CD63 and others that have a lot of CD63, for example, if this was the capture antibody. Would it be possible to have like a mix of capture antibodies, so you could have more capacity to for example, I was thinking in terms of biomarker discovery, because I would not like to limit my my exosomes to the ones that are very good expressions of CD63. For example, could I have like a mix of beads with different capture antibodies? Would this be customizable to this extent, for example?
This is something possible. Sometimes for some specific customer, we made some custom beads with a specific biomarker. It depends on the number of the beads you are planning to analyze. But I think that the first step probably is to try to with a general kit with CD, for example, for plasma, I recommend you CD9 for the capture beads and CD81 for the detection. This is the components of the plasma kit. And you have these beads are fluorescent for APC, but also for PCP and for APC's cyanine 7. So you have several biomarkers, several channels free, FTC, the violets, and so on. So the idea will be, in a first approach to a study, to try to capture the general exosome and to locate if this general exosome has the biomarker you are looking for. It has a very good signal for this specific biomarker, this specific marker. It's possible to change the combination and to use this antibody specific against this specific biomarker to coat it bits and to use these bits just to capture the specific exosomes you are looking for. I don't know if you understand me.
Yes.
So this.
Yeah, but to make a point, the CD9 will detect the exosome, so it will not be dependent in the amount or the antigen density. The 81, the mean intensity of fluorescence will be proportional to the amount of antigen density.
Yeah.
So one way to also interpret that can be, for example, if you have a spread, a CV, a big CV, what it means is that probably you have differences in the exosome expression. So you have a more heterogeneous population of exosomes in your population. A tight CV in the detection will give you a more homogeneous antigen density than a big spread.
Thank you.
Thank you, Nuno, for your question. There is, so any more questions? if not, we will finish this session. And not only the session, is also the webinar. So this is the last session. And I need to thank you all for being present, so all the speakers, thank you so much for sharing your knowledge and for contributing for this, and to know a little bit more about flow cytometry and how you can apply. In this case, it was about beads, as you know. So we talk about beads, and we try to make an overview about this subject. And today I need to particularly thank to André for sharing your knowledge and to talk about this. I think it's been like a hot topic, these EVs and the analysis of these particular particles, let's say. So these small particles has been, it's a challenge still. So the analysis of them is still something that we need to discuss a lot. And thank you so much, Ricardo, for being here today, and for also to share and to show how we can analyze these extracellular vesicles with beads. So, and of course, I always need to thank Alexandre Salvador for helping me in all the steps, and for always being with me with these challenges of organizing webinars or workshops. Is always helping me. So I need always to thank him. And so if you have any comments, any suggestions, because we are thinking to do more of these TraCyTalks with another topic, feel free to tell us what you thought about it, what we can improve. Will be really important to know your comments, and we are happy to know your messages. So I think you all know my email, so you can André is saying that he doesn't know. It's katrina.meirelles@i3s.up.pt. And so now, I will finish, and I want you to to turn on your cameras for the last time of this webinar, and to make a picture. And now I can see your faces, not only the names, or half of the faces nowadays. It's only the eyes mostly. So it's on. Okay, let me just put this in. Okay, full screen. Okay, are you ready?
I would like to thank Katarina also. You did a good job. If you need anything else from us, and I hope that we have contributed a lot to incrementing the knowledge and awareness of some of these teams. And we are open to suggestions. So next time you have ideas, count with us. We enjoyed a lot being in here. So if you need anything from us, just tell us. And I wish you all the best with the small things or big things, but especially with small things today. And obrigado, Ricardo. Gracias.
Obrigada, André. Obrigada a todos.
Merci, André.
Merci.